This is the next in a series of videos I'm doing on kind of basics surrounding bacteriology. And in this video, we're going to focus on bacterial growth, metabolism, and methods for sterilization. There are parts of this that as an entering medical student, you should definitely know, how quickly is bacterial turnover, how can you identify bacteria, and certainly how can you sterilize for bacteria. And then there are some concepts that are a little bit more niche. So I'll try to highlight those that are important and those that maybe you don't need to focus as much on. So the first thing is we need to think about how we're going to identify bacteria in general. And some of this we talk about when we talk about bacterial structure, but I'll just kind of reiterate it here. So the first thing is the shape. So you'll hear me talk about bacteria according to their morphology a lot. A typical description for an organism would be something like this: gram-positive diplococci. When you hear those words, you should automatically be able to form an image in your mind of small, dark purple little spheres that are grouped in pairs. And this particular description is, almost entirely used for the organism Strep pneumoniae, which is a major cause of obviously pneumonia, but also meningitis in patients. So we think about the shape. So the shape in this case is cocci. So there are three main shapes that we talk about: rods, spheres, and spirals. So rods would be bacilli, spheres would be cocci, and spirals would be spirochetes. And I'll point out more shapes in the structures video and as well as we talk about it. Size. Sometimes an organism will be described as very small or very large, but yeah, sometimes we can think of it that way. So for instance, if I had said small gram-negative cocci, the organism that most quickly comes to mind is a Neisseria organism because they tend to be on the smaller size. Some organisms are defined by their ability to move. Not all organisms can move, but some can, and they might do it in a particular way. For instance, Proteus is referred to as having swarming motility. When an organism can move, it has some structure that provides that. So be it either flagella or pili. There are also many organisms that are non-motile, and part of the way they move around the body is that they actually are invasive. They eat. Gram reaction. So I mentioned that here. So this is a gram positive. That means that it has that really thick peptidoglycan that stains really well using a gram stain. If it doesn't have the thick peptidoglycan, it becomes a gram negative organism. Okay, and then the last thing is macroscopic growth. So basically, how are we able to grow out this organism? What type of media works best? Does it require oxygen? What are its metabolic requirements? The other thing that I'll briefly touch on is ecology. Bacteria can be identified by their ecology, where in the environment they're found. For example, the organism Pasteurella multicida is a common complication of infections that result from a dog bite, while infections of organisms of the Clostridia genus typically result from penetrating wounds or ingestion of products from the soil, because Clostridia includes the organisms Clostridia botulinum or botulism and Clostridia tetani or tetanus. And this is because these two organisms exist in really different environments. As with other organisms, PCR has made DNA sequencing a mechanism for identifying bacteria, and that can be used to differentiate different strains. And while PCR is a powerful tool, it is not used to identify all organisms yet. And there still are a lot of methods that are used for culture and biochemical assays that are still being used to identify bacterial organisms. And we're going to learn those kind of throughout as we learn organisms and the tests that are used to identify them. So let's lay some groundwork for bacterial growth in general. First off, much like with parasites and other infections, the mere presence of a bacterial organism does not cause disease, which is good for us because we are actually covered inside and out with bacteria. We're actually more bacteria than human if you even just look at our gastrointestinal tract. And that's all because of the commensal microbiota that live throughout our body and provide protection to us. These are typically non-pathogenic organisms as long as they stay where they're supposed to go. A good example of this would be like E. coli. You've got E. coli all over your gastrointestinal tract right now, but you're not feeling ill, right? Hopefully. I hope you're not feeling ill. If that E. coli, say, went to your urinary tract instead, it doesn't belong there. And if it replicates there, it will cause a problem. So mere presence of the organism is not enough to cause disease. The organism needs to be found in a specific location. The other thing is bacterial growth is often required to cause disease. In most cases, it's the growth of the bacteria that is the cause. In a few instances, ingestion or injection of a bacterial toxin will cause disease. A good example of that would again be botulinum or tetany, where it's their toxins that are leading to disease, but for the most part, it's growth. And treatment with antibiotics is actually looking to do one of two things: either kill the organism, which is bactericidal, which obviously reduces the bacterial burden, or be bacteriostatic, which basically just slows the growth of the organism until your body can get rid of it itself. We can measure bacterial growth in 4 phases in the lab. There's the lag phase, the logarithmic, the stationary phase, and the decline phase. This is kind of a gross representation from a YouTube video that I really liked on bacterial growth. So basically, when you inoculate a culture in the lab, you have what's called the lag phase. This is basically where the organism is asking, where am I? It's getting used to the culture medium, the temperature, where it's at, and getting ready for this phase of rapid growth. Once it adjusts, it's going to grow exponentially, and that's the logarithmic phase. During this phase, the bacterium undergoes rapid cell division, and the culture is at its highest viability. Cells divide largely by binary fission, in this stage allowing for this exponential increase. This phase will last until culture nutrients become limited or toxic metabolic products produced by the bacterium as it divides inhibits its growth. It's this stage of growth that is typically responsible for rapid onset of disease, like say bacterial meningitis, is when the organism is growing rapidly. Stationary phase. When the culture gets too crowded, the organism stops dividing as much. And this is partially because we start to see a limitation in the amount of nutrients present. So during this phase, the rate of cell division is equivalent to cell death. So the viable number of bacterial cells remains constant, and that's what's being shown here. The colony will undergo some adaptive changes to survive. For example, some organisms, like the Clostridia and Bacillus species, might begin to undergo sporulation, which basically is them saying, I'm going to create some escape pods for when these nutrients run out. Then we enter the decline phase. During this phase, the nutrients have all been used up, the toxins have built up. This is no longer a friendly area to be in. And the rate of death is basically going to depend on the exhaustion of the nutrients, the temperature, the pH, the oxygen, and moisture within the culture. Some bacteria can survive for long periods of time on moist materials, so think like soiled bandages and bedding, unless the materials are sterilized. So that's why we still have to worry about sterilization, even when it would appear that nutrients would dry up. So part of the way we found these 4 stages was basically by inoculating cultures in the lab and figuring out how fast things were growing. And to do this, we needed to be able to measure bacterial growth. And we can do this in a couple of ways. The first one is turbidity. This is where you use a spectrophotometer, and it will basically tell you the total bacterial cell count, including all dead and live cells. But it's not going to differentiate between the live or dead cells. It's just going to tell you how many bacterium are present within a culture. So if I look at this, it's going to look at how many of these dots are basically scattered in this test tube, but it's not telling me how alive or dead they are. You can also do a direct particle count using a microscope or electronic particle counter. Again, this will tell you the total bacterial cell count, including all live and dead cells, but it doesn't differentiate between them. The only way to really differentiate between them is with some sort of viability assay. So like a viable cell count using colony forming units can be done. In this case, one colony is formed from one live bacterium. So if I streak this plate, right? This blood agar plate, and I see this. That means when I initially put this on here, there were 1, 2, 3, 4, 5, 6, 7 colonary forming units. So initially there were 7 live bacterium. This is the only method for differentiating between alive and dead cells reliably. Generation time. This is another thing we can do, and it's basically, once again, you're seeing that same curve that we just talked about. So here's your lag phase, here's your logarithmic phase, here's your stationary phase, and here's your decline phase. And we can figure out how quickly it takes a bacterial culture to double by basically using 2 points on the log phase. So if we start at 4 log number of visible cells and then go up to, I don't know why they did 7, it should be 8. So you look for a time of doubling, and then you count the number of hours in between the doubling. So it's fairly easy. You just identify 2 points in the log phase, which represent doubling of viable cells on the y-axis, and read the time on the x-axis. And that's going to basically tell you the generation time for doubling your culture. As I've already mentioned, the bacterial metabolism vastly affects what we see in our growth curve, right? Because as there's a lot of nutrients, we see a lot of growth. As nutrients begin to run out, growth slows, and when nutrients are gone, growth goes away and cells start dying. So it's profoundly affecting the metabolism and the environment. And this is because in a pure culture, there aren't any specialized cells to maintain the environment. So within our bodies, monocytes and macrophages come in and they clean up dead cells and they remove toxins, and organisms can keep going. But in a pure culture, that's not going to happen. So there are physical factors that are associated with how a bacterial culture will grow. The first would be things like temperature and pH. For most organisms, a temperature of 37 degrees Celsius, or basically body temperature, is ideal. pH, you want to stay pretty neutral, around 7. So these are just factors that's kind of where a bacterial culture likes to live. Nutrition, they've got to eat something. So you need carbon sources, amino acids, vitamins, inorganic ions. Most bacteria require a water-rich environment with these kind of preformed organic compounds for energy and a carbon source. They're going to require amino acids, purines, pyrimidines, vitamins, but the amount is going to vary from organism to organism. Similarly, there are some organisms that are halophiles, love salt, and they need a really salty environment, or need a high amount of magnesium. And so you're going to need to think about that when creating your laboratory media, which is kind of the third component. There are a lot of different types of medium, and I will kind of try to list them out as we go. At minimum, a laboratory media has to have water, glucose, and minerals. Sometimes a nutrient-rich broth is used, which normally will contain some sort of meat extract or hydrolysate of complex proteins. And bacteria are able to switch to using the compounds to them during the lag phase. So that's part of why we see that lag phase, because they're kind of identifying what's the food source here that I'm able to use. There are also different types of selective media. So selective media means that the media has been altered in such a way to help grow out a particular type of organism or indicate something. So selective media might favor growth of a particular organism. So an example of this would be like Thayer's marten. So Thayer's marten is actually fairly selective for Neisseria species, while it has antibiotics that will inhibit antibiotics that Neisseria is largely resistant to. So it normally contains something like vancomycin or something. You can also have indicator or differential media. This is media that kind of helps tell you what the organism is as it's growing. So an example of this would be McConkey agar. So McConkey agar contains lactose. And there are some organisms that when they grow on McConkey, they will turn the media a pink or red color if the organism is able to ferment lactose. E. coli is one of these. So when it grows out, we know that we're dealing with a lactose fermenting organism. As I mentioned previously, the minimum laboratory requirements are just glucose, water, but we also need to be able to provide oxygen. Okay, so bacteria produce energy from glucose in order of different efficiency, either fermentation or anaerobic respiration or aerobic respiration. All right, so we're going to have more efficiency as we get toward aerobic respiration. Not all bacteria are capable of all three of these different mechanisms. In fact, different bacterial species have different requirements for oxygen. Aerobic respiration can completely convert 6 carbons of glucose to CO2 and water plus energy. These organisms have enzymes called catalase and superoxide dismutase, which basically allows them to detoxify hydrogen peroxide and superradicals, which are byproducts of this respiratory process. Organisms that are unable to perform respiration don't have these enzymes, and therefore oxygen is highly toxic to them. So we actually have organisms that exist on this sort of spectrum based on how much they work with oxygen. So obligate aerobes, these organisms need oxygen to make ATP. They're only able to perform aerobic respiration. They're unable to do fermentation or anaerobic respiration. A good example of this is Mycobacterium tuberculosis, which most people have heard of tuberculosis. It's an obligate aerobe. Then we have on kind of the other side of the spectrum, obligate anaerobes. These guys can't do any aerobic respiration, so therefore oxygen is toxic to them. They'll perform fermentation instead. So they're going to perform glycolysis. In this case, glucose is going to be metabolized to pyruvate and then lactate or other compounds, which can be useful in identifying bacteria. This includes organisms like the Clostridia. An example of this would be like Clostridia difficile, also known as C. diff, which many of you have probably heard of. It's a nosocomial infection, which causes this severe, foul-smelling, watery diarrhea, typically in patients who have recently taken antibiotics. Okay, then you have this group. These are the facultative anaerobes. These organisms are living the best of both worlds. They can perform aerobic respiration, they can perform anaerobic respiration, or fermentation, depending on whether or not oxygen is present. This class represents the majority of organisms because they can kind of grow anywhere. Aerobic growth is typically preferred because you get more ATP for your effort than with anaerobic. A good example of this is E. coli. It grows just as well in your gastrointestinal tract as it does on your kitchen counter. So it's a really easygoing organism. Okay, then you have these guys, the microaerophiles. These guys need a little bit of oxygen. Too much is toxic. An organism from this group is H. pylori. We'll learn about this in your gastrointestinal tract. This is actually the cause of almost all stomach ulcers and cancers, and stomach cancers specifically. And it's microaerophilic. Then you have the aerotolerant anaerobes. They aren't going to do anything with the oxygen, but it doesn't bother them. These are able to metabolize or to colonize a wide range of places, much like the facultative anaerobes. They're just not using the oxygen. An example of this would be like S. pyogenes or group A strep. This is the organism that you have probably all experienced at some point in your life because it causes strep throat. So all of this is almost completely useless unless we know how to get rid of some of these organisms. Many of you will be in contact with a wide range of bacterial organisms over the course of your practice, and you need to know how to sterilize them. And obviously something all doctors typically do when they walk in and out of a room is to wash their hands, right? Because that prevents the spread. But there are a lot of other things we do as well. So the first thing is heat. Heat is a great way for getting rid of a lot of organisms. For most bacterial organisms, heating something to 100 degrees Celsius for at least 2 to 3 minutes will kill anything actively growing. However, there are some organisms that make spores. Spores are highly resistant to heat. The organisms most notably that make spores are the Clostridia, which I've mentioned a couple times in this video, as well as the Bacillus. Bacillus group contains the organisms Bacillus cereus and Bacillus anthracis. That's right, the causative organism of anthrax. Spores are really resistant, and you can kill them by steaming at 121 degrees Celsius for approximately 15 minutes. This is going to kill most of the spores. You can also use dry heat like an oven at 160 degrees for an hour. So you're going to need a higher temperature and longer time. There's also the process of pasteurization made by our friend Louis Pasteur. This is basically what we do to a lot of dairy products, right? So it's 62 degrees Celsius for 20 minutes. That's going to destroy most pathogens, but it's not going to affect the flavor of the product because it's not going to break down the proteins, and that's why we use it on milk. Okay, now what if you can't heat? What if heating it will actually destroy the solution you're trying to use, which is a common problem with laboratory media. You want to be able to grow the organisms. Eventually, you just want to grow the right ones, and so for now, you want it to be sterile. So in that case, we use filtration. For most organisms, if you have a 0.22 micrometer pore size, that's going to remove the majority of the bacteria that might be present from your solution. Okay, then we have disinfection. Disinfectants are basically chemical substances that are able to kill microorganisms on surfaces, but are far too toxic to apply internally. An example of this would be something like bleach. If you ever dump some bleach on some red blood cells, it will light up with bubbles the way if you dropped like some pop rocks into a Coke. Just big explosion. So bleach is a common one that people think of, but there are a lot of options. So alkylating agents, which is basically like something like ethylene oxide. The gas is used to sterilize objects that are heat sensitive. Formaldehyde, which is an embalming agent, is also a fixative that will work well for disinfection. Oxidizing agents, this would be like hydrogen peroxide. It can kill bacteria when used at 3 to 6% and spores at 10 to 25%. So it's a pretty good option. You can use iodine, which works well at a low pH. Iodine only works if the area is clear of organic compounds. So this is why if you go to like give blood or get surgery, they'll sometimes like wipe it down with like alcohol first to get rid of some of these organic, and then they'll use iodine. Chlorine compounds like bleach, a 5 to 10% solution is usually good for getting rid of materials that way. You can also use membrane disrupting agents. So these would be like quaternary ammonium compounds, alcohols. So for example, when we're thinking about alcohol, specifically most labs use ethanol, which is really good. Or you can use isopropanol. Interestingly, a 70% ethanol solution is actually more effective at disinfecting than a 95%. So more isn't always better. And then the last one would be like soaps and detergents, which we all use every day. There's all that antimicrobial soaps and detergents that you can use for sterilization.